Hello, hi everyone. My name is Dr. Sana Khan, your biology expert, and you're watching our app that is InstaPreps. So in this video, we are going to talk about the structure of antibody. Till now, we were studying that once the antigen goes inside, our immune cells produce antibodies. Now let us understand in detail how does this antibody look. Okay, so let's start and study about the structure of antibody. So you must have seen this structure before also in one of the videos, right? So this is how antibody looks. Can you tell me which shape is it? Yes, obviously it is Y-shaped structure, correct? And the nature of antibody is it is glycoprotein in nature. It is what? It is glycoprotein in nature. And who produce antibodies? Antibodies are produced by plasma cells formed by B lymphocytes. We have talked in the previous video. If you have not seen that video, please go to the app and do watch that video because it's a very important video. In that we have talked about each and every cell of our immune system. So plasma cells, which are also one of the type of lymphocytes produced a clone of plasma cells, and these plasma cells produce what? These plasma cells produce antibodies. So as I told you before, it is a Y-shaped structure which you can clearly see it in the diagram. Okay, now this Y-shaped structure is formed from 4 polypeptide chains. How many chains? Form 4 polypeptide chains. This is the first one. Then comes the second one. This is the third one. And this is the fourth one. So this is how 4 polypeptide chains are formed. Can we revise it again? Yes. So first polypeptide chain followed by the second, then third, and then this is the 4 polypeptide chain which form a Y-shaped structure. So it is a polypeptide chain. So it is made up of what? It is made up of amino acids. So these 4 chains are made up of amino acids, and this is a formula of amino acid. So what are the two most important part of it? That is the first one, which is amino group, and the second one is called as carboxyl group. So these are made up of amino acids, and amino acids have amino group as well as carboxyl group. So now one end of the polypeptide chain is made up of amino group, where I have written N. Can you see? So one side is made up of amino group and the other side is made up of carboxyl group, where I have written C. Okay. So one end amino group का है, दूसरा end carboxyl group का है, because amino acid के structure में दोनों भी group होते हैं. Now these are two. Can you see the two long ones? So the two long chains are called as heavy chain or edge chain. What are they called? Heavy chain or edge chain. And the smaller chain is called as light chain or L chain. What is it called? Light chain or L chain. So the bigger one, okay, the bigger one like this, which is like this, almost like this. Yeah, right. So the bigger one is called as H chain or heavy chain, and the smaller one, which is just like this, it is called as light chain or L chain. Now, the heavy chains have more amino acids, that is, their molecular weight is more as compared to the light chain. So light chain have lesser amino acid, that is lesser molecular weight. Now there are 4 chains. So there should be structure which could hold this 4 chain together very tightly. Yes. So the 4 polypeptide chains are held together by a disulfide bond. Can you see a disulfide bond between them? So these are the disulfide bond which helps to combine, which helps to hold this 4 polypeptide chain together at one place. See. And finally, these form together a Y-shaped structure. Now, the region holding arms and stem of antibody is called as hinge. So this is the arm and this is the stem, correct? So ye wala jo hai, that is your arm, and this lower one is your stem. So this region where my arrow is now is an area which is called as hinge. What is it called? Hinge. So hinge is an area which holds arms and stem of an antibody. Now, each chain will include 2 distinct regions. It will include what? It will include 2 distinct regions. So, the first one is called as outer region. So, outer region, which is also called as variable region. What is it called? Variable region. So, each chain includes 2 regions. So, this one is the outer one, which is called as variable. So the word variable itself says that it is going to vary because the word is itself saying its meaning. So the outer one is the variable region which keeps changing, and the inner one is a constant region. See this one in the heavy as well as light chain. So the inner one is a constant region which is not going to change. So each chain has two regions, variable which is going to keep on changing and the constant region which is going to be constant. Okay, it's going to remain same. Now this variable region, it is variable because it has antigen binding site, and we know there are n number of antigens going inside our body, right? So this variable region includes antigen binding site. Can you see? And that is called as paratome. What is it called? Paratome. So variable region constitutes the antigen binding site, this site, and that is called as paratome. So most antibodies carry two antigen binding site. Very, very important for MCQ. So most of the antibodies will carry how many antigen binding site? Two. See, the first star is the first one and the second one is the second one. And as it carries two antigen binding site, it is called as bivalent. What is it called? Bivalent. Why? Because it carries two antigen binding site. Now, let us just revise once again. So, 4 polypeptide chain, one end is amino group, the other end is carboxyl group. All the chains are held together by disulfide bond. The area which holds the arm and stem is called as hinge. So, there are two heavy chains and there are two light chains. Each chain has two parts. The outer one is the variable region which is going to change, and the inner one is a constant region which remains constant. Okay, so that is your structure of antibody. I hope it is clear with you. Okay, so now let us go to the next part of the video, that is antigen antibody complex. See, now we are studying from the first lecture itself that each antibody is specific for a particular antigen, right? And the antigen binding sites are located where? Now we have studied that antigen binding sites are located in variable region. See, so this is your antigen binding site. So now we are just going to study about this antigen binding site. We are going to take that antigen binding site over here. So this is the light chain and this is your heavy chain. Okay, and both the chains are of variable region only because this site is present only in the upper section and not in the lower section, right? So we are going to take only the upper section of the chain. So it's a one is the heavy chain, one is the light chain, which is here having an antigen binding site. See, this is your antigen binding site, which is called as paratome. So I told you that there are two sites. That is why it is called as bivalent. Okay, now let us understand what happens when the pathogen comes. So this is a pathogen. Okay, and this is a variable region. So variable region will enable to recognize that specific antigen. So it will analyze that antigen, it will understand it, and it will try to bind with that antigen. Can you see? I'll show you again. So this is your antibody. So antibodies has come to know, okay, now antigen has come here. So it is going to analyze that antigen and then finally it is going to get bind to it. See like this. Okay. And that once it binds to the pathogen or antigen, it forms a complex which is called as antigen antibody complex. What is it called? Antigen antibody complex. See. So antigen antibody complex is formed. Now we are going to just take this antigen and antibody complex. So this is the antigen. Now this site, can you see this one? This is a combining site of the antigen where the antigen is combining. And that site is called as antigenic determinant or it is called as epitope. What is it called? Epitope. So antigen ke upar, there is a combining site of antigen which is called as epitope. Don't get confused between paratope and epitope. So paratope is present on your antibody and epitope is present on your antigen. Okay, I hope that's clear because that's again an important MCQ. The difference between epitope and paratope should be very clear. Okay, we have understood that the variable region of antibody has an antigen binding site which is called as paratope. Antigen has an antigenic determinant antigen binding site which is called as epitope. Now, This variable region binds with a specific antigen in a lock and key manner. In a lock and key manner. Understood? So this epitope and paratope are highly specific because we have studied that antibodies are very much specific in nature. And study of antigen and antibody reaction is also called as serology. Is it clear? So I hope you have understood this part. We have talked about the structure of antibody in this video, about the 4 chains joining with the disulfide bond. Then we talked about the hinge, we talked about the variable region, we talked about the constant region. Then we talked about the 2 most important words which should be very clear, that is paratope and epitope. Paratope is present where? On the antibody. Okay, and epitope is on the antigen. Okay, in the next video, we are going to talk about how this antigen and antibody interact. Now we know that once the antigen comes with antibody, it binds with in a lock and key mechanism, right? So now when it binds, exactly what kind of reactions it shows, what kind of interactions it shows, that will be seen in that we will be doing it in the next video. Okay, so I hope you have understood. And again, as usual, I will tell you that if you have any doubt, do write it in the post to me. I will surely read it and answer your questions. Okay, till then, bye. Work hard, work for your dreams. I'll meet you soon with the next video.